Now I want to talk about the evolution of a healthy cell to a cancer cell. And I want to first tell you what it's not. So this is what it's not, but it's what sometimes I think people think. So I think it's important to talk about. It's not when you have it's not when you have a normal cell, or a healthy cell, I should say, that gains mutations, and you produce this cell here that is weak and dying. If that was the case, it might be easier to treat. But that's not what cancer is. That's not how cancers evolve. Rather, what happens is that you have this healthy cell that gains mutations and produces a cell that has a competitive, replicative advantage over neighboring cells. So you could have a situation where you have all these cells here that are doing their job, and then you develop one cell here that for whatever reason has gained a mutation or maybe several mutations that allows it to grow faster and outcompete its neighbors here for resources, eventually making it so that these previously healthy cells have a lower chance of survival because these cancer cells have a replicative advantage. They're going to take all the nutrients from these healthy cells. They're going to grow faster. They're going to push these to the side. They're going to make it harder for them to replicate. And then they're going to metastasize and colonize other areas of the body. Again, in those other areas of the body, they will have, again, that replicative advantage and continue to grow and outcompete their neighbor cells. So forming a cancer is certainly not forming a cell that is weaker or a dying cell. It's forming a cell that in many respects is much stronger than the other ones. Now, it's not necessarily a good strategy for these cancer cells because ultimately they kill the organism, and by killing the organism, they themselves cease to exist as well. Now, as they're going through this process of gaining mutations so that they can grow and outcompete their neighbors here, cancer cells will take on some features that I think we need to talk about that make them stronger and able to outcompete the healthy cells. And I've got about 7 features to talk about, so let's get moving on that. So let's talk about these 7 features. The first one is what I'll call self-sufficiency. What I mean by that is that they don't need external factors to tell them to divide or survive. So in a healthy cell, you're going to have these receptors that bind to things like a mitogen that tell the cell to divide. A cancer cell may still have this receptor, or they may not, it doesn't really matter, but they're going to divide, and in fact divide rapidly, independent of this receptor being signaled. So they're self-sufficient. The second feature is that they're insensitive to anti-growth signals. So similarly as here, but if you remember our cell cycle, where we had G1, we had S, G2, and M, remember we had these stop signals, 3 of them. One of them was at G1S, so let's just talk about this one. The cell may receive a signal that says we should not divide anymore. Maybe there's a mutation that the cell wants to repair before it divides, so the cell stops here. However, in a cancer cell, these stops are ignored. They don't respond to these anti-growth signals to tell the cell to stop, to fix a mutation before you divide again. Third, they evade apoptosis. A healthy cell may have various mutations in it, say lots of mutations, and the cell is determined it cannot fix it, so it signals apoptosis, and the cell begins to commit to the cell death pathway. And if you remember, that all started because of a gene called P53. When DNA damage is detected, it will activate P21. P21 will then inhibit cyclin CDK from phosphorylating the retinoblastoma protein, which is attached to E2F. Okay, so let me say that again. P53 activated because of mutations. It activates P21, which prevents cyclin CDK from phosphorylating RB. So it's not phosphorylated. As long as RB is not, retinoblastoma is not phosphorylated, the transcription factor necessary to drive the cell cycle remains bound to RB until the mutation is fixed. Once the mutation is fixed, this is reversed. E2F can now go do its job of progressing the cell cycle. Now sometimes P53, remember, will sense so much damage that it will cause apoptosis. And this is an important thing because sometimes the damage just can't be fixed. Like when someone is sunburned and your skin begins to peel. That is apoptosis. In cancer cells, one of the mutations that frequently occurs is a P53 mutation. Without P53, you can't make active P21, and so you can't inhibit cyclin CDK. So cyclin CDK is always phosphorylating retinoblastoma, and E2F is always moving into the nucleus to turn on genes to progress the cell cycle. Also, as related to this particular feature here, without P53, you cannot cause apoptosis that is triggered by P53. So this is how cancer cells evade apoptosis when they should undergo apoptosis. 4. Limitless replicative potential. This is sort of related to some earlier features when we talked about how the cell doesn't need signals to divide, it doesn't stop at the cell cycle checkpoints, and it allows certain genes to always be on to progress the cell cycle. But what I'm more specifically talking about here is the role of telomeres in cancer. So let me remind you what a telomere is. So we have our chromosome here, and our telomeres are at the ends. I'm going to draw them in purple here. And what usually happens in most cells, most somatic cells, is that as the cell divides many times and copies this chromosome, the telomeres get shorter and shorter by mechanisms that you learned about or will learn about if and when you take a genetics course. And at some point what happens is those telomeres get so short that it signals for apoptosis. A typical cell, human cell, will divide about 50 times, and it's linked to the size of the telomere. Now what happens in cancer is that these telomeres never get shorter. They continue to remain the same length after many divisions. So even when it's hit that limit of 50 cell divisions, it still keeps dividing. So the signal for apoptosis is not achieved through the shortening of the telomeres. So telomeres don't get shorter in cancer cells. Okay, the fifth feature I'm going to talk about. Cancer cells have a greater mutation rate and have more chromosomal abnormalities. So the mutation rates we've sort of talked about before. By gaining those extra mutations in various cells and in various genes will allow that cell to continue to evolve. And because it's blowing past those stop signs of the cell cycle, more mutations can accumulate. But something we haven't talked about is how the chromosomal structures change. In cancer cells, we see more translocations. That's when parts of chromosomes move to other chromosomes. We see more inversions. That's when a part of the chromosome is cut out and flipped around. Deletions, large segments of the chromosome deleted. Duplications occur more frequently. And something called aneuploids occur more frequently. And what that means is that instead of having 46 chromosomes, you will go from 46 to maybe having 45 chromosomes or 44 chromosomes. So you can get fewer chromosomes. Or you can get more chromosomes. You can get 47, maybe 48 chromosomes, or even more. So all of these occur at a greater frequency in cancers, which again helps them in their evolution of becoming that cancer cell that can move around the body and invade other tissues. 6. Cancer cells are invasive. Usually cells hold together nicely, and in most cases will form single layers like this. Some tissues, they form multiple layers, but there's a very nice structure to them because they are held together. And often they are held to some extracellular matrix on the bottom here. And if you remember from a few lectures ago, we talked about these proteins that help hold cells together, and these proteins were called cadherins, and they helped hold them together. And there were other proteins that would hold them to this extracellular matrix. It gave them this support network to allow the cells within a tissue to function properly. Well, cancer cells lose this ability. Their cadherins don't work properly. You'll still have your healthy cells here, but the mutated cells that don't have cadherins anymore will lose their attachment to each other and to this matrix below, and now they're free to escape this area and move to other parts of the body because they're no longer held together and they're no longer held to this tissue. Linked to the fact that they're invasive, they can proliferate in new locations. This is in part caused by angiogenesis, forming that blood vessel or recruiting the blood vessel to this tumor so that they can escape this primary site and travel to other sites and colonize to form a secondary tumor. We talked about the genetic nature and the mutations that can cause cancer and what those mutations do to the cells to allow the cancers to evolve, but we haven't talked about the genes themselves. And cancer genes can be lumped into two types of cancer genes. So types of cancer genes. The first one are tumor suppressor genes. Sometimes when people think of cancer genes, they think of these as new genes that people with cancer have. But that's not the case. Everybody has a tumor suppressor gene. And these tumor suppressor genes, when they're working properly, they stop cell growth or cell division. They are the ones that function at the G1S checkpoint, the G2M checkpoint, the spindle fiber checkpoint. They stop the cell cycle when the cell cycle needs to be stopped. Examples would include P53 that we've talked about and retinoblastoma, which we've talked about. So we draw our cell cycle over here. Tumor suppressor genes function here, here, and here at the cell cycle checkpoints. Now, when mutated, cell cycles checkpoints are ignored. So think of these tumor suppressor genes as the brakes of the cell cycle. And when they're mutated, the brakes can't be applied and the cell cycle is allowed to move forward. Tumor suppressor genes are what we call recessive genes. And if you remember from genetics or from an introductory biology course, recessive genes are ones where both copies have to be mutated. So these are our two chromosomes, and there is a tumor suppressor gene here, and let's just say it's retinoblastoma. If one of them is mutated, for the time being, that's okay, because this one here will still make the retinoblastoma protein, and the retinoblastoma protein, this one copy, is enough to serve as the stop for the cell cycle. Now, if they both become mutated, then neither one of them are making retinoblastoma, and this cell cycle checkpoint can be moved past without control. These mutations of recessive genes, of these tumor suppressor genes, are called loss of function. For them to have an effect that leads to cancer, they have to lose their function. Now I want to talk about the second kind of cancer gene, and these are called protooncogenes. Just like we said the tumor suppressor genes were the brakes of the cell cycle, we can think of the protooncogenes as the gas of the cell cycle. These are the ones that, once they pass the checkpoints, once the tumor suppressor genes have allowed the cells to move past the cell cycle checkpoints, the protooncogenes will drive the cell cycle forward. So they drive cell cycle forward. An example would be E2F. The tumor suppressor gene, RB, holds E2F until it's passed. Once the tumor suppressor gene has been removed, E2F is allowed to drive the cell cycle forward by transcribing genes necessary for S phase progression. When mutated, they ignore these stops. It doesn't matter what retinoblastoma is doing. So when mutated, they no longer respond to these stops. So maybe there's a mutation here that prevents it from binding to RB. So RB is still here, unphosphorylated, so it's trying to bind to E2F, but it can't because of certain mutations on the E2F protein here that prevents it from binding. In this case, it will always drive the cell cycle. And so when they're mutated in this way, we no longer call them protooncogenes, we call them oncogenes. These we refer to as mutations, or we could call them gain of function, because they're gaining a function of not responding to these typical signals, and they're just moving forward with the cell cycle. So we have these two chromosomes, and we have our protooncogenes on each of these, and we can say we're talking about E2F. And by various mechanisms, E2F or protooncogenes can be mutated, but they're not mutated by losing function. They're mutated in such a way that allows this gene to be expressed at a higher rate. So there are three ways we should talk about that protooncogenes can be converted into oncogenes. One is overexpression. For whatever reason, this promoter is allowing the gene to be expressed at a higher rate. Another way is that it could be duplicated. So we increase the copy number. So maybe instead of having just these two genes, we don't necessarily overexpress it from any one gene, but we create extra copies of it. And so every time it's turned on, in this case, instead of turning on two of them, we turn it on 4, which can be enough to drive it forward. And we can also have point mutations, where we don't have any changes in the expression or the copy number, but there is a point mutation. And when this point mutation occurs, it makes a protein that maybe, for instance, is unable to bind to retinoblastoma. If it can't bind to retinoblastoma, it will move forward. So you should know these three ways as ways of overexpressing a proto-oncogene and thereby making an oncogene. And another important feature I want to mention that's really important is that since it's dominant, this effect, this mutation here or the overexpression of it, only has to occur on one allele. This allele here can be completely unaffected. It can be behaving as normal as possible. But because it's this dominant mutation, only one of them has to be mutated or its expression changed in some way. Okay, that's all I have for the types of cancer genes, and it's all I have for this particular podcast. And sadly, this is also our very last podcast of the semester. It's been a lot of fun. I hope you enjoyed it, and I'll see you in class. Bye.